Hi everyone, this is Martin Tone, and in this video we are going to discuss about photosynthesis, mainly about light reaction in photosynthesis. Let us start with a brief introduction about photosynthesis. As you all know that life on earth ultimately depends on energy derived from the sun. Photosynthesis is the only process of biological importance that can harvest this energy. The term photosynthesis means literally synthesis using light. Light energy drives the synthesis of carbohydrates from carbon dioxide and water with generation of oxygen. When we discuss about photosynthesis, we are discussing about photosynthesis in higher plants. The most active photosynthetic tissue in higher plants is the mesophyll of leaves. Mesophyll cells have many chloroplasts which contain specialized light absorbing pigments which are green in color. They are known as chlorophyll. The chlorophyll appears to be green in color because it absorbs the lights of other wavelength and it reflects the green color. In photosynthesis, plant uses solar energy to oxidize water, thereby releasing oxygen and reduce carbon dioxide, thereby forming large carbon compounds, primarily sugars. Photosynthesis consists of two types of reaction. They are light reaction and dark reaction. Light reaction are those reactions which are dependent on light. That is, they require the energy from the sun or any other light source. They are also known as thylakoid reactions. The thylakoid reactions of photosynthesis take place in specialized internal membranes of chloroplast called thylakoids. The end products of these thylakoid reactions are high energy compounds ATP and NADPH. Dark reactions are light independent reactions, that is, they does not require the energy from the sunlight or any other light source. They are also known as carbon fixation reactions. Otherwise, they are also called the Calvin cycle. During dark reactions, they convert the carbon dioxide into glucose using the ATP and NADPH that was produced during the light reaction. Chloroplast is the site of photosynthesis. In photosynthetic eukaryotes, photosynthesis takes place in the subcellular organelle known as the chloroplast. The most striking aspect of the structure of the chloroplast is the extensive system of internal membranes known as thylakoids. Thylakoid contains the green light-absorbing pigment that is known as the chlorophyll, which is the major site of the light reactions in photosynthesis. Carbon reduction reactions, which are catalyzed by water-soluble enzymes, take place in the stroma, that is the region of the chloroplast outside the thylakoids. Most of the thylakoids appear to be very closely associated with each other. Their stacked membranes are known as grana lamellae. The chloroplast also contains its own DNA, RNA, and ribosomes. A wide variety of proteins essential to photosynthesis are embedded in the thylakoid membranes. As you can see from the image that chloroplast is a double membranous organelle which contains an outer envelope and an inner envelope. In photosynthesis, the light-dependent reactions take place on the thylakoid membranes. The thylakoid membrane contains some integral membrane protein complexes that catalyze the light reactions. There are four major protein complexes in the thylakoid membrane. They are Photosystem II, cytochrome B6F complex, Photosystem I, and ATP synthase. These four complexes work together to ultimately create the products ATP and NADPH. The light-dependent reactions begin in photosystem II. When a chlorophyll molecule within the reaction center of photosystem II absorbs a photon, that is, an electron in this molecule attains an excited energy level. Since this state, that is the excited state of an electron, is very unstable, the electron is transferred from one to another molecule, creating a chain of redox reactions called an electron transport chain. The electron flow goes from photosystem II to cytochrome B6F complex to photosystem I. In photosystem I, the electron gets the energy from another photon. The final electron acceptor is NADP. Thus, the light reaction continues. The electrons ejected from chlorophyll travel through a series of electron carriers organized in the C-scheme. The C-scheme of electron transport tells us how an excited electron gets transferred from photosystem II to photosystem I and ultimately leads to the production of NADPH and ATP. The four major protein complexes that take part in the C-scheme are photosystem II, cytochrome B6F complex, photosystem I, and ATP synthase. The functioning of these four protein complexes can be summarized as follows. Photosystem II oxidizes water to oxygen in the thylakoid lumen, and in the process it releases protons into the lumen. Second point, cytochrome B6F complex receives electrons from photosystem II and delivers them to photosystem I. It also transports additional protons into the lumen from stroma. Photosystem I reduces NADP plus to NADPH in the stroma by the action of ferredoxin and flavoprotein ferredoxin NADP reductase. ATP synthase produces ATP as protons diffuse back through it from the lumen into the stroma. In upcoming slides, we are going to discuss in detail about the four major protein complexes and how the electron transport takes place in the C-scheme. Let us discuss details about the photosystem II, that is P680. Photosystem II is also known as water plastocyanin oxoreductase. Photosystem II is contained in a multi-subunit protein supercomplex. In higher plants, the multi-subunit protein supercomplex has two complete reaction centers and some antenna complexes. The core of the reaction center consists of two membrane proteins known as D1 and D2. The primary donor chlorophyll, additional chlorophylls, carotenoids, Fearfightings, and plastocyanins are bound to the membrane proteins D1 and D2. The photosystem II reaction center, along with its antenna chlorophylls and associated electron transport proteins, is located predominantly in the grana lamellae. Photosystem II produces a very strong oxidant capable of oxidizing water and a weak reductant. Water is oxidized to oxygen by photosystem II. Water is oxidized according to the following chemical reaction, that is 2H2O upon oxidization gives 2 molecules of oxygen, 4 electrons and 4 protons. This equation indicates that 4 electrons are removed from 2 water molecules generating an oxygen molecule and 4 hydrogen ions. The protons produced by water oxidization are released into the lumen of the thylakoid. One electron carrier, generally identified as YZ, functions between the oxygen evolving complex and P680. Photosystem II consists of water splitting complex, that is the YZ, chlorophylls and carotenoid pigments, and a reaction center, that is P680, Fearfighting and 2 quinones. Photosystem II absorbs red light of 680 nanometer and is precisely absorbs only red light of 680 nanometer. As you can see in the C scheme that the excited electron in the photosystem II get transferred to next electron carrier, that is the Fearfighting. Fearfighting is a chlorophyll in which the central magnesium atom has been replaced by 2 hydrogen atoms. From the image, it is clear that the magnesium atom is replaced by 2 hydrogen atoms. This chemical change gives Fearfighting some spectral properties and chemical properties that are slightly different from those of chlorophyll. The Fearfighting then transfers the electron to the next electron carrier, that is the Plastocyanin. 2 Plastocyanones, Qa and Qb, are bound to the reaction center and receive electrons from Fearfighting in a sequential fashion. Transfer of the 2 electrons to Qb reduces it to Qb2-, and the reduced Qb2- takes 2 protons from the stroma side of the medium, yielding a fully reduced plastohydroquinone. The plastohydroquinone then dissociates from the reaction center complex and enters the hydrocarbon portion of the membrane, where it in turn transfers its electron to the cytochrome B6F complex. The cytochrome B6F complex is a large multi-subunit protein. It contains 2 B-type heme and 1 C-type heme. It also contains a risky iron-sulfur protein in which 2 iron atoms are bridged by 2 sulfur atoms. The correct mechanism of cytochrome B6F complex, or the precise way by which the cytochrome B6F complex transports the electrons and proteins, is not correct or not yet understood. But a mechanism known as Q cycle accounts for most of the observations. In Q cycle, the plastohydroquinone is oxidized and one of the 2 electrons is passed along a linear transport chain toward photosystem I, while the other electrons goes through a cyclic process that increases the number of protons pumped across the membrane. In the next slide, we are going to look into detail how a Q cycle works and what are its benefits. Let us discuss detailing about how the Q cycle works. Let us focus our attention on the first image, that is the first plastohydroquinone oxidized. In the linear electron transport chain, the oxidized risky protein, that is FeSR, accepts an electron from the plastohydroquinone QH2 and transfers it to the cytochrome F. The cytochrome F then transfers an electron to the blue colored copper protein known as Plastocyanin, which in turn transfers the electron to the photosystem I. In the cyclic part of the process, the plastosemiquinone transfers its other electron to one of the B-type heme, that is denoted as CYTB, that is a B-type heme, releasing both of its protons to the luminal side of the membrane. Thus, in the first step, 2 protons has been released into the luminal side of the protein, that is denoted as 2H+. Let us focus our attention to the second image, that is second plastohydroquinone oxidized. The B-type heme transfers its electron through the second B-type heme to an oxidized quinone molecule, reducing it to the semiquinone. Another similar sequence of electron flow fully reduces the plastocyanin, which picks up the proton from the stromal side of the membrane and is released to the B6F complex as plastohydroquinone. The net result of the Q cycle is that 2 electrons are transferred to P700. Second one, 2 plastohydroquinones are oxidized to quinone form. And third one is that, and 1 oxidized plastoquinone is reduced to the hydroquinone form. In addition to all of this, 4 protons are transferred from the stromal to the luminal side of the membrane. Due to this accumulation of the protons, there will be electrochemical potential difference across the membrane. This electrochemical potential is used to power the synthesis of ATP by the help of ATP synthase. Let us discuss about the next electron carrier, that is the plastocyanin. Plastocyanin serves as mobile carriers to connect the two photosystems. Plastocyanin is a small water-soluble copper-containing protein that transfers electrons between cytochrome B6F complex and P700, that is Photosystem I. The protein is mainly found in the luminous space. Thus, the plastocyanin acts as a connection protein between the Photosystem II and Photosystem I. Let us look into the next photosystem, that is Photosystem I, and its reaction center P700. It absorbs the far red light of wavelength greater than 680 nanometer. The Photosystem I reaction center complex is a large multi-subunit protein complex. A core antenna consisting of about 100 chlorophylls is a part of the Photosystem I reaction center, that is P700. The core antenna and P700 are bound by two proteins, namely PSA and PSAB. Additional electron acceptors include a series of 3 membrane-associated iron-sulfur proteins or bound ferridoxins, and they are also known as FES centers. They are FESX, FESA, and FESB. Electrons are transferred through centers A and B to ferridoxin, which is a small water-soluble iron-sulfur protein. The membrane-associated flavoprotein ferridoxin NADP reductase reduces NADP plus to NADPH, thus completing the sequence of non-cyclic electron transport. Photosystem I produces a strong reductant that helps in the reducing NADP plus, and it also produces a weak oxidant. Photosystem I contains a chlorophyll molecule A0 and a phylloquinone A1, which act as the electron transporters or electron carriers, and it helps in the carrying the electron to the NADP reductase. Light-dependent ATP synthesis, which is known as photophosphorylation. ATP synthesis takes place in light reaction by the help of an enzyme known as ATP synthase. The electrochemical potential difference that was created by using Q cycle is used as an energy for ATP synthesis. Let us look into detail how ATP synthase enzyme help in the ATP synthesis. ATP is synthesized by a large enzyme complex which is known as ATP synthase. This enzyme consists of 2 parts. First part is the hydrophobic membrane bound portion called CF0, and a portion that sticks out into the stroma called CF1. CF0 appears to form a channel across the membrane through which the protons can pass. CF1 is made up of several peptides and they are arranged alternatively. The major peptides that includes in CF1 are alpha and beta peptides. CF1 portion of the ATP synthase enzyme is the complex that helps in the synthesis of ATP. In CF1 complex, adenosine diphosphate, that is ADP, gets attached with another inorganic phosphate to form ATP. In this process, one proton is transferred from the lumen to the stromal site. Let us look into the overall summary of light reaction. The transfer of electrons and protons in the thylakoid membrane is carried out by 4 protein complexes. The water is oxidized and protons are released into the lumen by Photosystem II. Photosystem I reduces NADP+ to NADPH in the stroma via the action of ferredoxin and the flavoprotein ferredoxin NADP reductase. Protons are also transported into the lumen by the action of cytochrome B6F complex and contribute to the electrochemical proton gradient. These protons must then diffuse to the ATP synthase enzyme where their diffusion down the electrochemical potential gradient is used to synthesize ATP in the stroma. Reduced plastocyanin and the plastocyanin transfer electrons to cytochrome B6F and to PS1 respectively. Thus, by the light reaction, NADPH and ATP are produced.